Hey everyone, so today we're talking about mostly metabolism, but we're going to be hitting a couple things, macromolecules, enzymes, fermentation, lots of stuff. Let's get into it. First, I'm just going to go over briefly some of our big macromolecules again. We've talked about them a little bit before, but I want to hit them again because we're going to kind of encounter them a little bit. So we've got our carbohydrates or sugars. You can kind of use those terms interchangeably. If you have one single sugar, it's a monosaccharide. If you have two of them stuck together, it's a disaccharide. Multiple ones become polysaccharides. Of course, normally we depict them in one of two different ways. You either depict them linearly, like this, or you depict them, as you've probably seen it before, in a cyclic form. They normally exist in a cyclic form. It helps it stay stable. All those nice bonds, when they form this nice little circle here, it's much more stable than if it was broken like that, okay? But you can see this is where that bond would be. So if you start sticking them together, then you start getting your disaccharides. And there's three big ones, maltose, lactose, and sucrose. And you can see, just depending on which two monosaccharides you stick together, it changes what the actual thing is. And you can start stacking a bunch of these together. So you stick a bunch of maltoses together, or a bunch of lactoses, a bunch of sucroses, a bunch of glucoses in different orders and all kinds of stuff, and you can generate larger structures. So not only do we use sugars or carbohydrates as energy sources, but we also use them for structure. And so you may have heard of some of these, like just starch is another term that gets thrown around. So like starchy foods. This class and biochemistry courses are designed to dispel some of the notions. When you're trying to avoid starches or avoid sugars, well, you need to understand what that actually means. So like a starch is just like a large chain of carbohydrates or sugars, okay? It's not that different. So like a complex carbohydrate versus a simple carbohydrate, it's just how many sugars you got attached there. And how you actually branch those can change how they're stored. So like a basic starch, like an amylopectin, that would be a little easier to break the bonds between those individual sugars as well as amylose versus like a glycogen where you get a little more branching there. Cellulose would be just one of the many structural types that we have. So cellulose would be in like plant cell walls, like when you have to eat fiber, that's what cellulose is. And we do not have the enzymes to cleave the bonds that hold cellulose sugars together. We do have the enzymes to cleave the bonds that are starches. Or larger polysaccharides. So next we have fats. Fats and lipids can be used interchangeably. Generally, if you're in like a science course, you're probably going to say lipids. The better term would probably be fatty acids, though that can limit what you're exactly talking about. But in general, what you're looking at are really long chains of carbon. Okay, I'm actually going to jump ahead to the next slide here. So these long carbon chains with all these carbons going all along, and if it's saturated, that means it's just completely one long carbon chain with a bunch of hydrogens. It's unsaturated if you have a double bond between two carbons. And so if you're polyunsaturated, then you would have multiple double bonds. And so you could be monounsaturated, you could be polyunsaturated, but there's only one kind of saturated. Trans versus cis, so this would be cis because the hydrogens here are on the same side of the double bond. If they were on opposite sides, we would call that trans. So that's where you may have heard of the terms trans fat. So trans fat versus cis fats. We don't usually say cis fats, that's just natural fats will generally form cis-related double bonds, and so you get like a little kink right here. Trans fats look more like saturated fats, and that's why they're harder to break down. So they don't break down as easy, so that's why a lot of foodstuffs are made with trans fats, because they just don't break down. Whereas unsaturated fats, other organisms like to eat unsaturated fats because they look like every other kind of fat, whereas trans fats are kind of weird and don't look normal, and so a lot of organisms don't really like to eat those, but they have a longer shelf life. Which one's a liquid and which one's a solid at room temperature? Well, you can imagine you could stack more of these together than you could stack these because you get that little bend there. So unsaturated fats tend to be liquids at room temperature, so like olive oils and things like that, whereas like your butters are going to be more full of saturated fats. When I say fatty acids, I'm going to go back to this slide. So a fatty acid is where we have these long carbon chains. So these would be your nice long chains with your carboxylic acid on the end here. Okay, and so that's a fatty acid. It's the same here. So you've got this nice acid portion. So that's your carboxylic acid. When you form a bond here with like a glycerol, which is just a small little carbon chain here, we call those triglycerides because it can accept 3 fatty acid chains. And so that's another term that you may have heard of is triglycerides. Triglycerides are one of those ways that we store these long chain fatty acids for uses. And so that could either be for energy storage, you can use them to help make like your phospholipid bilayers. Fats are super duper important. So speaking of phospholipids and phospholipid bilayers are this general structure. They move around a lot. And you can see we've got our charged head here, and there's those fatty acid tails. And you can see they can be long chain, like so this would be a saturated fatty acid, and then you can get that double bond, and this would be a cis unsaturated fatty acid. And you can find those generally in healthier quote unquote foods. Moving on to the next topic is nucleic acids. So nucleic acids we've mentioned briefly before. Generally they're going to exist inside of a nucleus, though if you're a bacteria you don't have a nucleus. That's your DNA, your deoxyribonucleic acid, and your ribonucleic acid. We're going to talk at a later date about microbial genetics, so this will actually come up. But there are other nucleic acids. It's not just your DNA or your RNA. It doesn't have to be this long chain. You can use, basically if you take any of these bases, your A's, your T's, your C's, your G's, or your U's, and you can tack on like a sugar and then throw some phosphates on there, and now you have adenosine triphosphate. So you've got your base here, your nitrogenous base, because it's got lots of nitrogens in here. You have your sugar, so this is a carbohydrate, just like we just talked about. And then you can tack on these phosphates. And phosphates are really interesting. So it's phosphorus with lots of oxygens. And you'll notice normally you should only have 4 bonds max to something. And so the phosphorus here actually has 5 bonds, 1, 2, 3, 4, 5 with that double bond. Plus this oxygen has an extra negative charge. And so what's actually there is the bonds between these phosphates are incredibly energetic. They're relatively unstable. It's stable enough to maintain this molecule for at least a small amount of time, but you're able to store the energy inside these phosphates, especially the last bond here. And so if we break that bond, we're going to release a bunch of energy, and we're going to use that energy, and we're going to talk about that today. The last thing I want to talk about are proteins. So proteins are these long chains of amino acids or peptides, otherwise known as like a polypeptide chain. And those are what we generally think of as like your structural components, as in like your enzymes. The machines of the cell are going to have proteins in them. The channels that let things through membranes, things like that. In general, proteins have 4 levels of structure. The first level is called primary. That's just the order of the amino acids. There are 20 amino acids. There are some alternative amino acids, but in general, in most biological systems, the 20 amino acids is what we're usually talking about, especially like in humans. Though there are a couple extra weird ones. And so this polypeptide chain is just the order of those amino acids. Now, when you start folding them onto each other, you can form one of 2 general secondary structures. So we went from primary to secondary, and those are your alpha helices, or your alpha helix, and your beta pleated sheets, where they kind of fold over on each other. So an alpha helix actually kind of looks similar to DNA, that double helix. And beta pleated sheets are really like they look like little sheets. They're like these folded over amino acid chains. Tertiary protein structure is where we start folding the folds. And so we can take a couple alpha helices and fold them together. We can take a couple of beta pleated sheets and fold those on top of each other. And that's your tertiary structure. So it's folding the folds. Now, if you start taking multiple proteins of tertiary structure and sticking them together, that's where you get your quaternary structure. And so that's just multiple protein, multiple amino acid chains being combined together. One thing I wanted to mention is that you can take multiple molecules, multiple biomolecules, and stick them together. So for example, in this class we talk about peptidoglycan a lot. So peptido meaning peptides, glycan meaning sugars, and we'll talk more about the actual structure of it later. But you can see here we've got peptidoglycan, so this would be a really thick layer of peptidoglycan in a gram-positive bacteria, or a really thin layer in gram-negative bacteria. And you can see you've got these nice sugars, okay, just like we've seen. And then these would be your amino acids. They're not little circles or spheres or whatever, but that's just how they're depicting them. But that's a really, really cool mixed group of 2 different biomolecules. Jumping off of this idea of biomolecules, I want to start talking about enzymes. You've probably heard of enzymes before. Just FYI, I'm going to be cutting in snippets of a couple different videos to give you some visualization. I'm not going to introduce each time that I do it. Now, enzymes catalyze reactions. So what does that really mean? Well, let's talk about it. So first, let's go over the structure of an enzyme. So enzymes' main job, like I know there's a bunch of stuff going on here, but really enzymes' job is to grab onto something, which we call a substrate, and you're either going to cut the substrate in half and make two products, or you're going to take two different substrates and attach them together to make one product. Okay, so you're either going from one to two things to one thing. I mean, you could argue that you could do multiple things too, like, oh, I'm going to cut this thing 3 times and make 3 different things, but you get the idea. You're either cutting something or building something together. Now, they're usually made of proteins, but they have other biomolecules attached. They'll end in ASE. So just classic examples, pineapple juice is full of, and pineapples are full of proteases. So they end in ASE. Your DNA and RNA polymerases in your nucleus make DNA and RNA, respectively. And then reductases are responsible for causing reductions, which would be the gain of electrons, which we're going to talk more about later. The apo enzyme is like what most people would actually consider the enzyme. So that's in the gray here. That's just a goofy name that we give for it, but it's like the main structure of the enzyme. It's the main protein portion. And then you have these other cofactors, and these allow you to kind of control whether or not your apo enzyme is actually doing its job. There's tons of different cofactors. They get named funny names. I have a list in just on the next slide here, but if they're always there, we call those prosthetic groups. But most of your cofactors can actually come in and out, and they're going to change the catalytic site. So this is, sometimes it's called the catalytic site. More often it's probably called your active site. And that's just where your enzyme does its job. Okay, so all these things together combined will form your holoenzyme that has an active site. And that active site is going to do something, which I'm going to talk about that in a second.
Enzymes. Human body is a product of different chemical reactions and processes. But what controls these reactions? In 1833, a French chemist, Anselme Pien, was the first to discover the vital force that drove these reactions and named it enzyme. Enzymes are substances, proteins, or in some cases, ribonucleic acid, RNA, that speed up a biochemical reaction by modifying specific substances called substrates. Enzymes are supremely selective in who they bind to and modify, hence their specificity. Mode of action. Easier said, but how does a tiny enzyme speed up a chemical reaction? The answer to this is simple. Enzymes perform this critical task by lowering a reaction's activation energy. That is, the amount of energy needed for the reaction to begin. Enzymes bind to their substrates, hold them in such that chemical bond breaking and bond forming processes take place more easily. Enzymes have a specific place in them called active site, where the substrate binds and real-time action takes place. Active site has a specific size, shape, and chemical behavior rendered to it by specific arrangement of amino acids. Thanks to these amino acids, an enzyme's active site is unique only to a particular substrate. In addition to the active site, many enzymes consist of a non-protein part called cofactor. Cofactors may be cations, positively charged metal ions, activators, bound temporarily to the active site to activate the enzyme, organic molecules, vitamins or vitamin products, coenzymes, that join enzyme-substrate complex temporarily, prosthetic groups, permanently enzyme-bound. Many enzymes only perform their catalytic role when associated a coenzyme. An inactivated enzyme, protein, along with its coenzyme, non-protein, makes up a system called holoenzyme. To add to the complexity, scientists termed this inactivated enzyme as apoenzyme. And therefore, the equation becomes holoenzyme equals apoenzyme plus coenzyme.
Just to give you like a quick synopsis list of all the different kinds of coenzymes and cofactors that exist, you'll notice there's lots of metals down here. So these would be like micronutrients. This is why like you have to have zinc, you have to have magnesium, you have to have what's MO, molybdenum in really small amounts to actually allow your enzymes to do their jobs. There's way more coenzymes too. Some of these may sound familiar. We'll encounter acetyl-CoA today. We'll talk a bunch about flavonoadenine flavin adenine nucleotide and nicotinamide adenine dinucleotide, which sounds super big. They're not. It's just FAD and NAD. We're going to talk more about those later. They're like batteries almost, and they act as cofactors. And we'll talk more about what that really means later. So let's talk about enzymes and their general structure and just what are they really doing? Because you can get really verbose about enzymes and all their structures and all their coenzymes and all that crazy stuff. But basically, an enzyme just has an active site. It's got a spot where it's going to do its thing. It's kind of like having a tool, and only one part of the tool is the business end, and all the rest of it is just parts that allow that business end to do its thing. So like if you had like a table saw, the thing that actually does the sawing, so the blade that actually cuts the wood as you pull the table saw down, that would be like the active site. But there's a bunch of other components that are powering the saw, and that's all the other stuff in the enzyme. They're the things that give the enzyme its structure or allow the active site to have the specific shape that it does. You can see here, generally we depict enzymes in one of two different ways. This would be like what an actual enzyme looks like. I have another one on the next slide too. But you can see, you get this little binding site here. This is your active site. So that's where you can stick your little substrate in there, and it kind of sits in there nice and cozy. So let's talk a little bit about just that active site, just to kind of give you a visualization. So you've got your general protein structure. So this would be all the extraneous stuff. And then you've got your active site would be all of this area. It's actually like a darker gray, but it's really hard to tell. And then you've got where the things actually bind. Okay, so this would be the part that's going to hold on to whatever you're doing. So let's say that this was like a 2x4. This would be the blade that's going to cut the 2x4, and then this is your hand holding the blade or some other kind of holder thing. I'm not a carpenter, but you get the idea. So this is going to hold on to it, and then this does the actual action, okay? But they're both part of the active site. But you have a binding site and then the catalytic site. There are a couple different models of substrate binding. In general, the induced fit hypothesis is probably what's more true, but we actually can find both examples existing in nature, and so we need to talk about the two. There's lock and key, where just, you got your active site and your substrate fits right in there, does its thing, and then ejects it back out. The induced fit actually seems to happen more often because proteins, which enzymes are usually made of mostly protein, they can change their shape. And so maybe it can grab onto its substrate easier in this shape, but to actually cleave and catalyze it, it needs to change that shape. And so the actual presence of this in there allows this to enter a different state, and thus, so maybe like your catalytic site would be sort of inside the middle of this weird looking thing, and then it's allowed to cleave, but you can't actually access it until the substrate actually binds. And so that's induced fit.
Models of enzyme action. Lock and key hypothesis. This is the simplest model to represent how an enzyme works. The substrate simply fits into the active site to form a reaction intermediate. Just like the key fits in its specific lock, the shape isn't changed here. Rather, the structure of the substrate absolutely complements the structure of the enzyme, like puzzle pieces. Induced fit hypothesis. In this model, the enzyme, upon binding of substrate, changes shape. The matching between an enzyme's active site and the substrate isn't just like two puzzle pieces fitting together. Rather, the enzyme changes shape and binds to its substrate even more tightly. This fine-tuning of the enzyme to fit the substrate is called induced fit.
Enzymes in general have interesting kinetics, is what we would say. So how fast do they actually cut things? And they have preferences for temperature, pH, substrate concentration. It just depends. For a class like this, we're just going to kind of glance over this, but temperature is usually the one that we talk about the most. You can imagine if something's really cold, it doesn't really work as well. Let's say you need to cut a piece of paper and you're really cold. Well, you're kind of shivering and you just can't move as fast because your joints are all cold and it's just hard to move the scissors. And so as you warm up, though, you could start cutting paper in half faster and faster and faster. And then you hit a point where you reach your optimum abilities, okay? So you're cutting paper as fast as you can. I don't know why you're cutting paper, but you just are. And then somebody cranked up the thermostat. It's getting hotter and hotter and hotter. You're starting to sweat. It gets so hot that you catch on fire, and now you definitely can't cut paper anymore because you're dead. Well, that's how enzymes work. So as your temperature increases, the amount of activity that you can perform increases until you're dead. And so you actually see a rate of reaction curve that looks like this. So you're kind of ramping up your abilities until you hit your peak. and then you very rapidly, as you denature would be the term that we would use for an enzyme breaking apart. Because the bonds that are holding your enzymes, which are usually proteins, okay, so those beta sheets and those alpha helices, the bonds that are holding those pieces together to actually give you the structure to do your thing, whatever you're catalyzing, those can fall apart pretty easy as temperature increases. Another common issue for enzymes would be pH. So different enzymes function at different pHs. So pH is just how many hydrogens are floating around. Yes, it's that simple. So a pH of 7 would be, like a neutral, like just a straight up water. Everything's, it's in equilibrium, basically, the number of hydroxide ions and the number of hydrogen ions. And so some enzymes like working at that. So like enzymes in your mouth, so salivary amylase is what you use in your mouth that's going to cleave sugar. Your mouth is generally neutral. If you're heading into your stomach, though, your stomach has enzymes in it in addition to hydrochloric acid. And so the enzymes that actually exist inside your stomach like to function at more acidic pHs, which makes perfect sense because there's that hydrochloric acid is breaking stuff down in the food you eat, and you want the enzymes to work really well there too. And so they work really well at like a lower pH, like a 2 or a 3 in general. There are also certain enzymes that prefer basic environments. And so you can see that pH just can kind of control how well an enzyme actually works.
Environmental effects on enzyme function. Active sites are very sensitive. They sense even the slightest change in the environment and respond accordingly. Some of the factors that affect the active site and consequently enzyme function include temperature. The suitable temperature for enzymes to function properly is 37 degrees Celsius. Increasing or decreasing the temperature above 37 degrees Celsius affect chemical bonds in the active site, making them less suited to bind substrates. Higher temperatures denature enzymes. pH. Amino acids present in the active site are acidic or basic. Fluctuation in pH can affect these amino acids, making it hard for substrates to bind. Extreme pH values can denature enzymes.
Now, I want to talk just very briefly about how enzymes actually kind of work. This would be the biochemistry portion of this. So enzymes technically lower the activation energy of a reaction. What are they doing? Well, in practical terms, they're just bringing the reactants or your substrates or whatever you're trying to catalyze, you're bringing them close together and then promoting a reaction to happen. Okay? That doesn't mean the reaction will happen, it just makes it easier to happen. And so that's what we say when we say it's lowering the activation energy. So activation energy is just how much energy do I actually need to put into this to get it to happen? And enzymes make that easier by just... So a lot of times people will say enzymes lower the activation energy. Sure, that's true. That is what they do. But I'd rather you had a more practical understanding. What are they really doing? Well, they're bringing things close together. You've got your enzyme and your substrate, they bind, and it just makes things happen easier. They are a catalyst. They allow a reaction to happen at a lower activation energy that's more attainable in a biological system, as opposed to when you don't have an enzyme, then it's just not going to happen. So if you take a biochemistry course, or if you've taken a biochemistry course, we actually talk more about delta G, delta H, delta S, and some of the math that goes into actually studying this energy and what does it actually mean to lower your activation energy. But for a class like this, just understanding that enzymes make it easier for a reaction to happen.
To speed up a reaction in the absence of enzyme, additional energy would need to be provided as heat, which jostles the substrates and occasionally provides enough energy to trigger a reaction. In the course of most reactions, an unstable and highly energetic transition state is formed as the substrates are transformed into products. An enzyme acts as a template for the reaction, binding to its substrate and holding it in the proper position to form the product. An enzyme also surrounds its substrate with reactive groups that stabilize the transition state, making it easier for the reaction to occur.
So we know that enzymes have a specific structure, right? They have an active site and they have other structure as well, but that active site is what we generally think about. Now, you can have enzymes that are doing something, but sometimes you don't want them to do something, and that's where inhibitors come in. So inhibitors are just that. They inhibit the reaction. So if at top, Shout out to Khan Academy too, because I like their drawings. So we've got our normal reaction here. Here's your enzyme. There's your active site. You bring your substrate in and you're cleaving it and you're making it into two different products. Okay, awesome. If you want to inhibit that, well, there's in general two different ways that you can do that. So a direct or competitive inhibitor would just bind into the active site. I mean, it's directly competing with the substrate. So you got your substrate here, normally it wants to stick in there. You got your competitive inhibitor, it's just competing for that space because it matches up with the actual shape. And that's generally true. Your competitive inhibitors will at least have a portion of themselves that can fit into the active site. A non-competitive inhibitor, I was taught as an indirect inhibitor. An allosteric inhibitor would be the better term. Those bind somewhere else. And so a classic question is, what's the difference between a competitive inhibitor and a non-competitive inhibitor? Well, a competitive one binds to the active site. A non-competitive binds somewhere else. What does that do? Well, binding somewhere else, somewhere allosterically, somewhere else on this enzyme, in general, what happens is it changes the shape of the active site. Because the enzyme is like one big thing, and so if I start kind of pushing on one side of it, it's going to change this inner shape. And so it should show that the substrate's not actually binding in there, but they're just going to show that no reaction happens. So it can actually occur either way. So sometimes you can have the substrate fit, but it's not able to catalyze, or sometimes you could have it closed off so that the substrate can't even fit in there at all. Something else to think about is that it could be a reversible inhibitor or an irreversible inhibitor. So if you had a competitive inhibitor that was irreversible, that means once it's bound to the active site, it can never leave. If it was a reversible inhibitor, well, then maybe it could fall back out, and then you could have your enzyme keep doing its thing. But the same thing can happen for non-competitives. So if it binds here permanently, that would be irreversible. If this thing could fall back off, your inhibitor, then sure, yeah, that would be reversible. A lot of toxins or poisons are irreversible inhibitors. So we're going to shift gears now, and we're going to move into the main crux of what we need to talk about today, which is metabolism. So metabolism is a combination of anabolism and catabolism. So when we say metabolism, we're really supposed to be kind of summarizing all the anabolic and catabolic reactions that are happening. In common use, when you say metabolism, like, oh, I have a really strong metabolism, what you're actually saying is catabolism, the breakdown of something. So if you're breaking things down, that is catabolism or catabolic. If you are building something, we call that an anabolic process or anabolism. The key factor here is ATP. So we've talked about that before. ATP is adenosine triphosphate. It's a nucleic acid. It's the main energy molecule that we use to move energy back and forth between systems. It's what the cells use as their energy currency. There are other forms of currency that we will talk about, but in general, you're always aiming to get to ATP. It's the dollar bill of energy. You can have bigger bills, you can have 20s or hundreds or thousands, but I mean, you're not going to go to the dollar store and spend $1,000 bill, right? They don't even want that. What they want is a single dollar bill, and that's what ATP is. It's really easy to spend. So we need to talk about ATP, and we're going to spend most of today talking about how we make ATP and what catabolic processes we use to make ATP that we can then use in anabolic processes to make stuff. So let's jump to this diagram here. ATP we have here in the center, so adenosine triphosphate. Okay, so it's an adenosine with a ribose and then 3 phosphates stuck onto it, and ADP is adenosine diphosphate. And so on the next slide here, I have our ATP. So this is the adenosine with the ribose here in the brown thing. And you've got 3 phosphates attached. And if you recall, I said if you break off that end phosphate here, breaking that bond releases a ton of energy. And we can use that energy to do things. Once we've broken that ATP, though, it becomes ADP, adenosine diphosphate. There's 2 phosphates. And a PI, that stands for an inorganic phosphate. It's just the result of this. The phosphate has to go somewhere. And so you could argue that this is an unpowered battery. Now, technically, this bond also has energy, but we're going to kind of ignore that for this class. So you have your ADP, and you have that inorganic phosphate, and we need to rebuild our battery. And so that's where catabolic reactions are going to come in. Okay, so our catabolic reactions are going to take energy from somewhere else and rebuild this bond to make ATP. So I'm going to back up to this original slide. So if we're going to take our ATP, we can do anabolic reactions that way. It's going to release energy and heat, which I guess heat is a form of energy, but we're using that energy to build bonds for something else. So ATP is sort of sacrificed, that last phosphate bond. We sacrifice that bond in order to basically put that bond somewhere else. And we can make more complex things. But we can take some of those more complex things and break those down, and that would be called catabolism, and we use that to rebuild that last bond. So let's talk about how we actually make ATP. So ATP is made in 3 different ways, okay? It depends on what you're doing. In general, we're going to be talking about substrate level and mostly oxidative phosphorylation. I will touch on photophosphorylation. That's what plants do, so that's like photosynthesis. And so let's jump into it. Now, the first thing we need to talk about are redox reactions, which I usually get a reaction from students when I say redox reactions. All we're talking about is reduction and oxidation. So in general, if I am taking electrons, okay, so in this case, simple case, we're taking sodium, we're taking an electron, sticking it onto fluorine. Sodium was oxidized because it lost an electron. And fluorine gained an electron, which electrons we sort of arbitrarily assign a negative charge to. And so it gained a negative charge, and so it's more negative, and so its overall charge was reduced. So the gain of an electron is a reduction. The loss of an electron is oxidation. Why am I talking about this? Let's talk about this in terms of ATP. So if I have ATP, and I'm going to break this bond and release energy and send those electrons somewhere, that would be an oxidation of ATP and a reduction or gain of electrons for something else. And the opposite is also true. If I have my adenosine diphosphate and I want to build a new bond here, I have to take some electrons to sort of fill that gap and build that bond. So in that case, ADP is being reduced because it's gaining electrons to build that bond. And so if it's reduced, then something else was oxidized, okay? If you have one, you have to have the other. So if my ADP gets reduced to become ATP, something else was oxidized. And that's what we're going to talk about. What is getting oxidized? Usually when we're talking about what's getting oxidized, we learn based on glucose. Glucose is the simplest sugar that we can really tackle. It's got 6 carbons, and that's what we're going to be oxidizing. It could be other molecules too. So any kind of molecule that you can burn for energy, because burning things is basically oxidation, then anything that you can burn, so fat, protein, sugars, anything that you can burn is oxidizing. So if you're going to burn some fat, you're oxidizing that fat. And if you're oxidizing something, you are therefore also reducing something. And so we're going to be reducing this guy. So I mentioned earlier that there are 3 different ways that we actually make ATP. The technical term for that would be phosphorylation of ADP. So I'm taking adenosine diphosphate and I'm attacking on another phosphate with a bond. That's called phosphorylation. And there's 3 different ways that we do that. We're going to start talking first about substrate level. Substrate level phosphorylation is where an enzyme is going to hold on to 2 substrates. So we just talked about enzymes, right? So enzymes have their active site, which has a catalytic portion, and it's got a binding portion, so it's going to hold on to 2 different substrates, and we either break it or we make a new bond. And so what we're going to do is kind of both in this case, but in general, we're going to hold on to ADP, and there's our phosphate, and we're going to cut this bond and sort of stick it back on over here. Okay, so that's substrate level phosphorylation because our ADP and our phosphate, which is usually held onto some kind of carbon-based molecule, that's what's going on here, they're acting as substrates. And I'm going to be bringing up that oxidation and reduction happens every single time. So in this case, we would be oxidizing this carbon molecule that has the phosphate, and we're reducing ADP to become ATP, right? Because electrons have moved from this molecule onto this molecule. I have another picture of this same kind of process occurring here. In this case, this is the final step of glycolysis, which we're about to talk about. We're not going to go into it in too much detail, but we're going to talk in general about glycolysis because glycolysis is the quintessential process that occurs in both bacteria, archaea, eukarya. All the domains are going to use glycolysis to take a basic sugar and cut it into pieces and end up with pyruvate, which is this product down here, which is a 3-carbon molecule. And we're going to make ATP via substrate level phosphorylation. So we're going to use our enzymes to hold on to these two substrates, and then we're going to oxidize this one to reduce this one, right? So the electrons are moving over here, thus reducing ADP to become ATP, and thus we're oxidizing phosphoenol pyruvate to become pyruvate. The names aren't important. What's happening is what is important. So let's talk about glycolysis a little bit. So in glycolysis, we're oxidizing. Okay, so we're breaking bonds, releasing some energy, releasing some electrons. So if we're oxidizing glucose, so there's our nice simple glucose molecule, simple is a relative term, I suppose, we need to reduce something else. And so in general, we're going to be reducing ADP, we're giving it some energy and electrons and making ATP. And so that's what's happening here. So we've got an ATP being made here and another one being made here. This is the exact same step that I just showed you. This step is this step. Now, we release a lot of energy when we actually oxidize a glucose molecule. We cut it in half. We send each half through this process. So we make 1, 2, 3, 4 ATP from this whole process. But we also take some of that excess energy and we use that to build a bond between NAD plus and we make an NADH. One of the most important things to think about, and this is how it's usually couched, is you have to spend money to earn money. So you can't earn a bunch of money unless you spend a little bit of money on anything. So let's say you're trying to get your degree. Well, you need to pay money to a university to get your degree so that you can get a job and make more money. Biological systems are the same way. So in this case, we're going to take glucose and we can't just willy-nilly cut it in half and oxidize it. So what we need to do is spend a little bit of energy to actually do that process. And so that's where you see we have 1, 2 ATP at the beginning of the process that we can allow to break the glucose. And then eventually we cut it in half and we have it go through here. And so we actually make 1, 2, 3, 4 because we split glucose in half and send each half through this end of the glycolysis process. And so we actually end up with a net total of 2 ATP because I had to spend 2 to make 4. So in the end, we're making 4 ATP, 1, 2, 3, 4. We had to spend 2, so you have a net gain of 2. And 2 NADHs, 1 for each time that each half goes through here. I also want to just briefly show what these things look like. So there's ATP, okay, there's that super high energy bond right there. Here's NAD+, and look at this hydrogen right here. So we're going to tack on another hydrogen, so we're adding a bond, and bonds are energy. So we have to take some energy out of this process and use it to form this new bond here. So we're taking NAD+ and NADH. Another molecule we're about to encounter in the citric acid cycle is FAD. So we've got FAD here, and you can see where we tack on 2 hydrogens. And so we've got a hydrogen here with a bond, and we've got a hydrogen attached to this nitrogen and a bond, and those bonds have energy in them. And so later on, what we're going to do is we're going to break these bonds. So if we have ATP, we break this bond to get the energy out, and we can then perform anabolic reactions. In this case, we're going to break this hydrogen off, take that energy out, and use it through what's called the electron transport chain to make more ATP, which we can then break this bond and use it to do anabolic reactions. Same with FADH2. We're going to break these bonds to release energy and actually make even more ATP. So ATP is what we're kind of ending up as. So NADH and FADH2, you could kind of consider like, this would be like a $100 bill and this would be a $50 bill, and we're trying to break them into ones, and because ones are easier to spend. So just briefly, we need to talk about the citric acid cycle. So if you take a biochemistry course, we go hardcore into all the enzymes. So each of these steps is actually carried out by a separate enzyme. So every time you see a stop at an arrow, that's a different enzyme that's performing a function. But I want you to get the general idea of what's happening here. So acetyl-CoA is what came out of that pyruvate, which left out of our glycolysis process. So pyruvate, we lose another carbon, we gain another NADH because we broke a bond and then we made another NADH with it. And so that's where we're coming from. And then that acetyl-CoA actually gets smashed into an oxaloacetate. So you go from a 1, 2, 3, 4 carbon plus 2 makes a 6 carbon citrate. Okay, the names I'm not concerned about, but you're going to then just continue to break bonds using enzymes as you go through this whole cycle. So this is called the citric acid cycle. The citrate is citric acid. It's also called the tricarboxylic acid, or it's named after the dude who discovered it, who was named Krebs. So this is called the Krebs cycle as well. I like citric acid cycle because it's like the actual name here.
Fun fact! The Krebs cycle is also known as the citric acid cycle because of this very byproduct. However, it is usually referred to by the name of the man who figured it all out. Hans Krebs, an ear, nose, and throat surgeon who fled Nazi Germany to teach biochemistry at Cambridge, where he discovered this incredibly complex cycle in 1937. For being such a total frickin' genius, he was awarded the Nobel Prize for Medicine in 1953.
So if we're breaking lots of bonds, we're getting energy out. And so we can make all those cool little $50 bills and $100 bills or really big batteries, if you will. And we're going to eventually use those to make more ATP. And so you can see we make some NADHs here. We make an FADH2. We actually end up making a GTP or an ATP. It depends on the kind of cell type that you're actually in. But basically like a really easy to spend energy molecule. And that's using enzymes to do all of this. This is still all substrate level phosphorylation, at least in the case of making ATP or GTP. The next thing we're going to do is actually take these NADHs and FADH2s, which they're not phosphorylated, they're not gaining a phosphate like ATP is, but they are storing energy for later. And so let's see where they're headed, okay? So we have substrate level phosphorylation from glycolysis that's generating ATP, awesome. We have our citric acid cycle, which is also making ATP via substrate level phosphorylation. Now look up here. So we've got our NADH and FADH2, and those are bonds between those big structures, so NAD plus or FAD, and their hydrogens. And there's electrons and energy stored in there, and we're going to take those and we're going to send them to one of the coolest series of machines ever devised, the electron transport chain. And we're going to do a process called oxidative phosphorylation. The electron transport chain is responsible for oxidative respiration or oxidative phosphorylation, okay? It's actually really simple. All these things sound super cool. Proton motive force, hydrogen ion gradients. I'm gonna tell it to you straight up, okay? You're gonna take your NADH, you're gonna take your FADH2, and you're gonna pull those hydrogens off, okay? You're breaking that bond. And when you rip that bond apart, you take the hydrogen that's left over and you stick it on the other side of this membrane. So in this case, we're in the mitochondria and we're between the outer membrane and the inner membrane on the intermembrane space. And it's a nice hollow area that's just full of hydrogen ions. And then you've got that electron, and instead of sending the electron over here, because that would balance out the charge, we keep the electron stored in what we call the electron transport chain, okay? Because we want all these nice positive hydrogens on one side of a membrane. If you'll recall, if you put a bunch of anything on one side of a membrane, we call that a gradient. So if we have a high concentration of something on one side and a low concentration on the other side, all the things on the high side really want to move towards the area that's lower. That's a concentration gradient. What we've actually made is what we can call a proton, because these are just hydrogens, they're protons, motive, i.e. it can cause power, it's a motor force. And so having all these hydrogens on one side, we're going to allow them to pass through this special machine called an ATP synthase. And it almost acts like a water wheel. So if this electron transport chain is what allows us to put water on one side of a dam, and then we can allow that water to come back out on the other side of the dam, and we can use that to generate power. In this case, we're making ATP. It really isn't that different from like how a dam works, where you could, or like a water wheel, where you could put water or whatever you want on one side of it and allow it to pass back through, and that movement allows you to power something. We see hydrogen gradients all the time. These occur in mitochondria. These also occur inside bacteria. So in a gram-negative bacteria, it's really easy to think about because they would have like a thin layer of peptidoglycan here, but then they have two membranes. Gram-positives are a little trickier, but basically they would use their peptidoglycan as their outer layer here and put the hydrogens in between. It's not as well understood how they actually perform this process, but this is where that occurs inside of a bacteria. So inside of your cells, inside of your mitochondria, which are inside of your cells, this is where that occurs in between that, in the intermembrane space between the two membranes. In a bacteria, it occurs in their outer membranes. A really important point to hammer home here, you'll notice the electrons, they're passing through this electron transport chain. So we're left with these molecules here that we can then have go back through in the process. We have hydrogens on one side, but those hydrogens and those electrons, they need to go somewhere. They can't just sit there. And so we need to send them somewhere. And so the hydrogens are going to pass through. And so we're going to end up with these hydrogens on one side of the membrane, back on this side. And then those electrons need to go somewhere. And so what we do is we build a new bond or a new set of bonds using oxygen and those hydrogens. So we take an oxygen molecule. It's saying O2, but just imagine if there's one oxygen. And so we have an oxygen molecule, and oxygen likes to have two bonds, okay? So we're going to take one hydrogen and bond it to the oxygen. And we're going to take another hydrogen and bond it to the oxygen. And we have now just made an oxygen with two hydrogens, otherwise known as H2O. So oxygen in this context is what we call our terminal, meaning at the end, electron, because our electrons are kind of flowing through and hitting this, acceptor, because oxygen is accepting those electrons. So oxygen is called a terminal electron acceptor, at least in the context of the electron transport chain. This is why you breathe oxygen. It's the terminal electron acceptor for your electron transport chain, which I know is a mouthful, but the terms aren't as important as understanding the actual process of what's happening. You got to send your electrons somewhere, so let's send them to oxygen. That's why we breathe oxygen. Oxygen is amazing at grabbing onto electrons. We talked about this very early on in this course, that oxygen is almost the most electronegative atom that exists. Fluorine is technically more electronegative, but oxygen is like the second most electronegative atom. This is why it acts as such an awesome terminal electron acceptor, because oxygen is super electronegative, which means it really likes electrons. It wants them so bad. And so we use it in our electron transport chain because it's going to act as an awesome electron sink, if you will, to actually grab onto this. Kind of like a heat sink absorbs heat. Oxygen can absorb electrons super well, and we can actually use it to take these hydrogens sort of out of the mix, and we make water. And so that's how we can actually kind of finish this whole process. Because oxygen is so electronegative, it's used in all kind of combustion/oxidations. So in this case, you could just take like a sugar cube and light it on fire, and that's instantly releasing all of the energy inside that sugar cube. Or another thing you could do is we used to take we'd have a bonfire and we'd throw potato chips on it and they just kind of explode almost. Or you can take a marshmallow just full of sugar and throw it onto a fire and it just kind of burns and all that energy comes off. It's really bright, right? So all of the energy inside of that sugar is being released in the form of heat and light. Well, in this case, what we're doing is we're doing what we call a stepwise oxidation. If we lit something on fire, that's like an instantaneous burning or instantaneous oxidation. If we do it stepwise, we can take a little bit of energy out of every step. So an enzyme is responsible for each of these steps. And we slowly pull the energy out of that sugar. In the end, we end up with the exact same products. We're still ending up with carbon dioxide because the carbons in the sugar had to go somewhere. And we end up with water, which is sort of holding on to the oxygen that we had to use from our initial combustion or cellular respiration or initial oxidation process of sugar. So I'm going to cut to this crash course video because I really like how it visualizes what's happening.
So remember all those NADHs and FADH2s that we made in the Krebs cycle? Well, their electrons are going to provide the energy that will work as a pump along a chain of channel proteins across the inner membrane of the mitochondria where the Krebs cycle occurred. These proteins will swap these electrons to send hydrogen protons from inside the very center of the mitochondria across its inner membrane to the outer compartment of the mitochondria. But once they're out, the protons want to get back to the other side of the inner membrane because there's a lot of other protons out there, and as we've learned, nature always tends to seek a nice, peaceful balance on either side of a membrane. So all of these anxious protons are allowed back in through a special protein called ATP synthase. And the energy of this proton flow drives this crazy spinning mechanism that squeezes some ADP and some phosphates together to form ATP. So the electrons from the 10 NADHs that come out of the Krebs cycle have just enough energy to produce roughly 3 ATPs each. And we can't forget our friends, the FADH2s. We have 2 of them, and they make 2 ATPs each. And voila, that is how animal cells the world over make ATP through cellular respiration.
So if we back up real quick, back to this process, we know that oxygen is our terminal electron acceptor. Not all creatures on the planet need to use oxygen as their terminal electron acceptor. So we need to talk about alternative terminal electron acceptors. If you're an aerobe, that means you use oxygen as your terminal electron acceptor. So animals are aerobes, humans are aerobes, lots of bacteria are aerobes. But if you use something different from oxygen as your terminal electron acceptor, You're still using the electron transport chain. It's almost the exact same process. You're just kind of swapping out something else right here. Those are called anaerobes. Anaerobes can use a variety of terminal electron acceptors that are different from oxygen. I have a list up here. Lots of sulfur compounds. So if you remember back to your periodic table, sulfur is also relatively electronegative, as is nitrogen. And so it kind of makes sense that they would be decent terminal electron acceptors because they're relatively electronegative. However, oxygen is still always the best. So you can look over here. On this chart, we have our redox potential, which is in millivolts, which is a way of just measuring how much energy you can actually get out. And aerobic bacteria, so Pseudomonas being one of them, which includes you too, aerobic bacteria can have the most energy come out because they're using oxygen as their terminal electron acceptor. And then you can also measure the amount of energy that comes out in kilojoules or kilocalories if you're in the United States, though the numbers are technically different. But you can see these different terminal electron acceptors are generally relatively electronegative. And so that is nice because then I can send my electrons to them and allow this process to keep on happening. If you use either oxygen or a different terminal electron acceptor, we call those facultative anaerobes. Okay, so they're anaerobes in that they can use an alternative terminal electron acceptor. Facultative is sort of like, well, if they have oxygen around, then they're going to use that because oxygen is definitely the best. However, if there's no oxygen around, like they use it all up, then they will switch to something else. Now, what happens if you run out of your terminal electron acceptor? That's a good question. That process is called fermentation. Fermentation is not anaerobic respiration. Okay, that's a common misconception. A lot of people think that if you're an anaerobe or if you're undergoing anaerobic respiration, that means you are undergoing fermentation. That's not true. I want to dispel that now. Fermentation means you're missing the terminal electron acceptor. So if you're an aerobe and you don't have any oxygen, you perform fermentation. If you're an anaerobe and you're missing one of your like sulfur compounds or whatever you use as a terminal electron acceptor, you undergo fermentation. Glycolysis is still occurring because glycolysis doesn't care as a process if there's oxygen or not. So glycolysis is often called an anaerobic process. I don't like that term because it implies that oxygen is somehow related maybe to an alternative terminal electron acceptor. It's not. It's just sort of a misuse of the term of anaerobic versus aerobic. Okay, so I don't like referring to glycolysis as an anaerobic process. So glycolysis is still occurring. You're still going to make a little bit of ATP, right? Because we make 4 ATP when we do glycolysis, but we have to spend 2, so you have a net gain of 2. But you're also going to make NADH. That's another byproduct. That NADH is a problem because as that builds up, we're going to be using up our stores of NAD+. And we have to have NAD+ in order to act as a substrate for our enzymes to actually keep doing this whole process. And so if you're not restoring your NAD+ or making your NAD+ from your NADH, this whole process grinds to a halt. That's bad. And so we have this alternative process called fermentation to actually get rid of those NADHs and make them NADs. And so we have to get those hydrogens off of these NADHs. In general, there's 2 ways. I'm going to show that there's actually other ways to do that. But for the most part, usually we either talk about alcohol fermentation, which is where we take our pyruvate, which is the end product of glycolysis, make acetaldehyde, and turn that into ethanol. Okay, so that would be what yeast does to make beer or root beer or whatever you want. If you are doing lactic acid fermentation, which is what humans do, then we would take our pyruvate from glycolysis, and in order to get rid of those hydrogens, we send it using an enzyme, and we make lactate. And so, or lactic acid. So lactic acid fermentation, lots of other creatures can do it too. We just don't engage in ethanol fermentation. We don't have the enzymes to do that. So we do lactic acid fermentation. And lactic acid is what you build up in your muscles. That's probably something you've heard of before. The buildup of lactic acid makes it really sore. And so you need to get rid of the lactic acid before you can actually keep on doing your stuff. And so that's fermentation. There are other pathways of fermentation, just to kind of overwhelm you with a crazy chart. You can see here's the lactic acid fermentation, here's your ethanol fermentation, but there are lots of other things that you can ferment. So you can make different kinds of alcohols, you can make acetate, and I actually have another slide here that talks about some of the different things that you can make. So if you want to make cheese, that's pretty classic. You could make other kinds of cheeses using lactic acid. So using lactic acid bacteria is generally what you would use. Yogurt would be a big one for that. That was like my grad work, was using lactic acid bacteria. Of course, you can ferment things and make wine beer. Root beer is just like early beer, basically, with lots of sugar in it. You can make acetone or isopropanol, like rubbing alcohol. You can make acetic acid, which is vinegar. So you can do all kinds of cool stuff. But the key here is that you're taking NADH, pulling those hydrogens off and putting them on something else. And so you see lots of hydrogens. How do we get those hydrogens onto something else? Well, we usually just tack them onto some other kind of carbon molecule. So we're nearing the end of the talk. I want to reiterate a few things. So the idea of catabolism. So we've been talking about basically a bunch of catabolic processes. A question that sometimes comes up is that breaking ATP into ADP and an inorganic phosphate energy and using that to do something. So the doing something, so like building a protein or whatever, that is an anabolic process. And so we have to break ATP, which is catabolic, so because we're breaking something. And so we're breaking ATP to make something. So how are those two things different, right? Anabolic and catabolic is generally reserved for big macromolecules. The breaking or forming of ATP is generally ignored when we talk about if it's anabolic, making ATP, or if it's breaking ATP, i.e. catabolic. We don't usually use those terms for ATP because it gets too confusing. So we're generally talking about the bigger process. And you can see on this chart, there's a combination. You can take any molecule and either break it down, i.e. oxidize it, and thus reduce things eventually to become ATP, or you can go the other way and build something and do an anabolic process. There are so many different pathways. If you take a biochemistry course, you generally will tackle these things. But for the most part, just understanding that if I want to make a protein, I can either break it down or I can build it. Same with DNA, same with all of your biomolecules. Fats, sugars, they can all be generated, i.e. anabolically, or they can all be catabolized to generate energy. So I glossed over earlier that there are 3 ways that we actually make ATP. Just to be a completionist, there was substrate level phosphorylation, there's oxidative phosphorylation, which is your electron transport chain. If you are like a plant and you have chloroplasts, you can do photophosphorylation, which is almost identical, sort of, to how we actually do oxidative phosphorylation. So you'll notice there's a lot going on here, but I'm going to use energy from the sun. So there's the light hits these photosystems, photosystem 2 and photosystem 1. That gives us the energy to actually put hydrogens on the other side of a membrane. And so if I put hydrogens on one side of a membrane, I'm making a gradient. And if I make a gradient, I can use that to power, oh, look at that, ATP synthase, just like what was in our electron transport chain. So that's kind of cool. So that's the 3rd way, but for the most part, we're generally talking about substrate level and oxidative phosphorylation. One thing I just need to cover really fast is how we classify organisms. These terms may sound familiar, but we just need to make sure that they're super clear. So where do you get your energy from? Well, that's a good question. So if you use the sun, you're a phototroph. If you use anything else, you're a chemotroph. Okay, so when we're doing breaking glucose down using our mitochondria, and like you're a creature that if you're not a plant and you need to eat something to get that energy, you're a chemotroph. If you use sunlight as your energy, it's a phototroph. If you need to get your carbons, well, I usually think about humans. So humans, like I'm going to eat like a candy bar and there's a bunch of glucose in there. Well, I'm a heterotroph because I'm getting my carbons from glucose. If you're an autotroph, then you're sucking it in from the atmosphere. So that would be like a plant would be getting CO2 from the atmosphere. Something that doesn't get talked about too much is where you get your electrons from, because usually it's the same place. So if you're a heterotroph, then whatever you eat, i.e. you're a human eating a candy bar, your electrons come from the same place your carbons come from. There are certain creatures that can get their electrons from minerals. So I like to think about like sodium or potassium that will readily give up their electrons. And those are called lithotrophs. As you can see, they're just not even on this chart, but it's important to talk about because they come up in some classes. The last thing I wanted to do was just talk about how much ATP do you actually get out of the burning of one glucose molecule? Well, that's a really good question. Just to kind of end on a picture of what's happening, just to remind you, you had to spend 2 ATP to end up with 36 ATP net. So technically you make 38, but you had to spend 2, so I said 38 minus 2. That's using either substrate level phosphorylation plus oxidative phosphorylation via NADH and FADH2. I really like this chart because it just kind of summarizes everything that we've talked about. This is sort of a best case scenario. You don't actually get this kind of yield from one glucose molecule, more often because you don't necessarily take, like, your pyruvate doesn't have to go into acetyl-CoA. It can go off and do all kinds of other things. But in general, this is about how much ATP you would get. Now, you would also get less ATP if you use a different terminal electron acceptor, because oxygen is the best terminal electron acceptor. It has the largest free energy that you can actually gain out of it. And so if you switch up your terminal electron acceptor, well, then you would actually get less ATP per glucose. But you're still getting a lot more than substrate level, which is only 2 net glucose. And with that, I want to thank you for listening. That's how we get energy in a cell, be a bacteria, an archaea, a eukarya. We do almost all the same kinds of processes, though sometimes we change up our terminal electron acceptor. But in the end, we're always making ATP.